Hello and welcome again to Multibiomics. With this video, we are starting with our lecture today, and this will be a theory class because I firmly believe that as a bioinformatician, you should know about your data from where it has come and what it looks like and what type of data are present in it. So, the knowledge of statistics and being familiar with the data, these two things are very important as a bioinformatician. So that is why let us see what actually is single cell analysis and how the data is prepared and how you're getting those files for analysis, right? So let's start and we'll commence with our presentation. So the overview of single cell RNA sequencing. What is this? Single cell RNA sequencing, it is a method, okay, it's a protocol that analyzes the gene expressions, okay, profile of individual cells. Obviously, similar technology was there earlier, like real-time PCR or qPCR, right, or bulk RNA sequencing. But what happened was, like, suppose there's a tissue, and in that there are so many cells, okay, and when we take out the RNA and we convert them into cDNA library, and the expression profile that we got was the sorry overall average expression profile of all the molecules, all the RNA molecules. So, but single cell analysis, what it does is that we can get the expression profiles of all the single cells separately. So how does it help? It helps us to explore the variability in what are the other in like variability of different gene expressions that are present in our data. So why are these important? So variability in gene expression or different types of cells present in a tissue, it is termed as heterogeneity. So in tissues, cells are often heterogeneous and their expression can vary significantly among each other and also from time to time. Okay, so depending on the whether a person is suffering from a disease or if a person is healthy, so on basis of that, the gene expression profile also changes. Now, bulk RNA sequencing, what it did, since it gave us the average expression profile, it masked this variability, and we couldn't, look through the single expression of different types of cells that there are present, and thus it obscured some very important and valuable biological insight. So this is a just a flow diagram, so we can use animal samples or human samples, and then we can, using different platforms and technology, we can separate those cells and we can get different population of cells. So like here from the spleen, you're getting B cells, monocytes, natural killer cells, okay, PDCs, macrophages, and here when you're using human tonsil and small intestinal cells, you get different types of populations of the cells. And then we can proceed further with the analysis. So mostly we will be using fluid-based method. There's another method known as plate-based method. We will not go deeply into that method. A fluid-based method, what are the strengths? It is cost efficient. Sequencing of cell can be done in larger quantities. Okay, and we can identify the overall composition of tissue. That the tissue is made up of what different kind of cells. Okay, and we can even characterize some rare type of cells that are, their whose number is very less. So we can characterize such type of cells also. Now, unique molecular identifiers can be incorporated. these are nothing but these are the unique sequences that are tagged to the cDNA library. Okay, so the cDNA. So each molecules when they are expressed and amplified, every molecule, one RNA molecule, just a second. Okay, so this one RNA molecule, it had a unique UMI. UMI suppose 1, and the second RNA molecule had UMI 2. So when they are amplified, their copies will also carry their UMI code. Okay. Now UMI is nothing but the sequence. They are in the form of sequences only. Now suppose if you don't have UMI, we can get false expression levels, because if some RNA levels are some RNA are expressed highly, so what do we interpret is that the original expression level for that RNA molecule was high. But it can also be due to amplification, right? Because we are doing PCR. So with the help of amplification, we can increase the number of RNAs or cDNA libraries. So these UMI helps us to keep track of the original number of RNA molecule that were present in the cell, and our analysis doesn't get biased because of the amplification. Because if amplification happens randomly and if we do not identify those things, our analysis can be biased, and we would come to a conclusion, a false positive or a false negative result, that this particular RNA is expressed highly or uh, unusual or highly positively correlated or negatively correlated expression. Like these biased results we can get, but this UMI, unique molecular identifier, can solve this problem. But there are some limitations for fluid-based method. Obviously, fluid-based method, it means it will incorporate a liquid phase, right? Or a gel phase. So um, we can quantify more number of cells, but the detection rate of the transcript is less. Okay, and depending on the platform, it does not, it does not capture whole sequences. Okay, I'm having some sort of issues in my pen, but I hope will cope up with that. So the whole read is not being sequenced. You have got 3' end and you have got 5' end. So only little sequences that are near to 3' or only those little bases that are near to 5' end can be sequenced regarding depending on whatever kit you are using. Some kits allow 3' end and some kits allow the sequencing of near 5' end. Okay. So like 10x genomics in that Chromium technology. So Chromium allows us to sequence the bases near the 3' end. Okay, so moving further, yes. So this is just the workflow, like you have got some different population of cells and you are, dispersing them in the liquid base, and then you see here, you are encapsulating them in a gel, and then you are running your sequencing. Either you can perform single cell RNA sequencing, or you can perform bulk RNA sequencing. So in single cell RNA sequencing, you get the data for each and every cell's expression profile. But here you see you are getting an average single expression profile, and we cannot make out that what are the different populations of the cells that they are present in this sample, right? So the procedure of RNA-seq wet lab procedure, it begins with tissue dissociation using enzymes like protein digestive enzymes like trypsin and papain. Okay, so what they do is they break the cell addition and cell breaks free, and we get single cells. So cells are then counted for viability. That means cell must be live, and also the cell must be healthy, right? Only then we should load them into a specific chip. We have chip system, either in a plate based or in fluid based. We have got chip system. Okay. Now in that we also have some gel beads. Now these gel beads, they form a different phase. So this gel will be in a liquid phase here, and they can float, right? And each gel will have a single cell inside it. The formation of gem, okay, the it is kind of an gel-like emulsion. Okay, emulsion is very critical. Now, this is the most important step because in each gem bead, you have got a single cell. So, in plate-based method, you have got wells, and inside the wells, you will get the cells. But here, in fluid-based method, in the fluid, the gels will be suspended, and in each gel bead, you will have your cells. Okay. So this is the diagrammatic explanation. So this is the tissue, and when we use the digestive enzymes like papain and trypsin, so the addition is lost and we get single cells. Then after the cells are separated, it passes through cell sorter, and the cells which are not yet separated are sieved out through a mesh. And here we can also, with the help of the cell, we can count the cell. Okay, here the help of cell sorter, and then we usually go for multiplexing. Now, what do you mean by multiplexing? Because in one single aliquot, we have got different types of population. Okay, not a single population, but we'll have different population. So going for the further analysis, okay, going for further analysis, um in each well or in each gem, you have different populations of the cell. Now, since we are performing a single assay on different population, that's why we are known as multiplexed. Okay, I hope you understand. If you don't, you can just give me a query and I'll try to explain to you further. Now, so this is a, the apparatus. Okay, so it's filled with liquid base, and here it is a single gem emulsion where you have got a single cell. Here, these are the single cell. Okay, now so the cells and the gem beads, they come from here. The gem beads come from here, and the cells come from here, and they combine, and then they make this cell and gem emulsion. So when you look deeply into a single cell, you see here there are so many population of cells, and each cell is actually inside a gem. Now, how the bead looks. So, bead is tagged with a set of sequences where you have a primer that will be attached to your gem bead. So, here there will be primer, and then you have a barcode. This is a barcode. Now, barcode is a unique sequence. Sorry, it is a unique sequence. What it tells that this RNA came from which cell type. Okay, so barcode is a unique sequence. Here it will be attached, and suppose there are this yellow cell, suppose this is NK cell, and then this is B cell, and this is macrophage. All the macrophages, there will be many macrophages. All the macrophages will be represented by a single barcode. All the NK cells will be represented by another barcode. So by looking at these barcode, we can come to know that which cell type that RNA molecule has come from. So it will help us in annotation further downstream in the analysis part, right? Now UMI, when we come to UMI, I've already told you UMI is like when several RNA are amplified, so the amplified copies should not be counted as different RNA molecule. They should only be considered as copy of the single RNA. So this is to reduce the bias. And then obviously you have a poly T tail, because whenever an mRNA is coming, they are flanked with poly A tail, and this poly T tail will be providing the binding site for your RNAs that they are coming, right? So cell type will be um, identified with the help of this barcode sequence. So here you see primer is attached to your gem bead, right? And the same diagram, we go again. This is primer, and here you have got barcode and then unique molecule identifier. So when this particular mRNA is amplified, it will be amplified with the help of same UMI. The same UMI will be copied, and we'll come to know that these 3,000 copy or 30,000 copies are not separate RNA molecules, but they are just the copies of this RNA molecule, and the expression level will not be biased. And the poly T tail will be serving as the binding site for the poly A tail of our mRNA. Now, the main tags on the gem beads are, I have put over here. You can go through that. And we also have adapter sequences, which helps in library construction. Okay. Okay. Right. You can go through this table later. And this is the workflow that I've already told you. So many different population of cells are present. And here different cells are enclosed in the gem bead, and we are collecting it, and then we are attaching the barcode. Okay, then we remove the oil. Okay, and then we get sequencing ready libraries, and we put it into a sequencer. So there are some strength and limitations of fluid based method. So the first one is it is cost efficient. Okay, and then the cells in larger quantities can be studied, okay, identified, and we can also characterize the rare cell types. So these three advantages are there for fluid based methods. And then the fourth and the most important one is that the UMIs can be incorporated. But what are the limitations is that low detection rate of transcripts. There is not much amplification, so that is why there is a low detection rate of transcripts. Only 3' end or 5' end, only those bases, those who are near to the ends, can be sequenced, and that too according to which kit you are using. Now, you can go and look at the strengths and limitation for plate-based methods. Now, whatever method, either fluid-based or plate-based, there are several challenges in the data analysis part, because many messages or the signals will get lost, because many of the RNA molecules may get degraded or damaged, or they may even not come out of the cell, right? So these are known as dropouts. Okay, then even if you are getting some signals, this one will give you no signal. Here means most of the signal are lost completely. But what is this? Some part get distorted. That means if you are getting signals, but those will be noisy signals. So here you're getting noise. That's why we have multimodal approach. Okay. So what do you mean by unimodal? So it's a normal distribution. Okay, so these are the statistical part. So always consider your RNA-seq data as a multimodal data. You cannot treat this as a regular RNA data, right? So it is much more complex. Your single cell RNA is much more complex data set, right? So what do you mean by multimodal? Multimodal means you have got multiple patterns. A single gene will be expressed differently. Suppose if you have P53 gene, okay, so it will be uh, expressed differently in a normal tissue or a normal cell or differently in a cancerous cell, right? Because its function is different in different times or in different cells. This is just the summary table. So, okay. But we can also go for not only single cell RNA-seq, we can also go for single nucleus RNA-seq. So, single nucleus RNA-seq involves isolation of the nucleus, okay, instead of whole cells. Okay, now the nucleus are tougher than the cell membrane. So the RNA molecules are intact in even in the frozen tissues, right? And they can be extracted even without the use of enzymes. So when one of the advantage of single nucleus RNA is that it avoids biases because whenever the cell is in harsh condition, the cell may rupture or it gets disassociated, but the nucleus may remain intact and those RNA molecules can be taken for further analysis. So studies have shown that the nuclear RNA will also accurately reflect the cellular gene expression as equally as done by the cellular cytoplasmic RNA, right? So the choice between single cell and single nucleus sequencing, it depends on your tissue type, right, or the quality of the sample. So tissue type, we have seen whether it is frozen or very old sample, right? And the quality, if the quality is not good, then you go for nucleus part, right? Don't take single cell RNA. And also you must understand that the dissociation method can also be biased. How? Because different cell types are captured, right? And different cell types, some cells will be tougher, some cells will break easily, so there will be some bias. The cells that are breaking easily, those will have more number of RNA in the beads, and the cells that are tougher to break, lesser amount of RNA are released. So we must be aware of such biases. Collaboration between, that is why the wet lab collaboration and the dry lab collaboration is very important, and that is why I have taken this theoretical part. Okay, so these are some things that you can go through on your own and you can explore. So this is the end of my presentation, and I hope that most of the concepts got clear, and I wish you all the best with this particular course, and let's start with the practical from the next session. Thank you and have a nice day.